Hi Dan, so with photosynthesis, just a quick run through. Obviously we know the equation is carbon dioxide plus water with sunlight goes to glucose and oxygen. A reminder that water comes in the roots as luminal ions, no water comes in the leaves at all. Carbon dioxide diffuses into the leaves via the stomata, goes up to the palisade cells by a process of diffusion, and the sunlight obviously comes from the sun. And leaves are broad to absorb more sunlight and thin for a short diffusion distance. We need to know about the structure of xylem, which takes water up the plant, which has lignin impregnated into the sides to make it waterproof, and the end walls have died. And so when we have a plant like this, we have things that will affect the rate of photosynthesis. And they are carbon dioxide, temperature, mineral ions, and light intensity. And specifically on your question, we have 3 graphs for each of those that we need to know. So if we look at this first one here, light intensity. So with light intensity, we can see that as the intensity of light increases, the rate of photosynthesis goes up until the chloroplasts are saturated. And then you can see at this point here, it doesn't matter how much you increase the light intensity, the rate of photosynthesis doesn't increase. Okay, so we say therefore that light intensity is no longer a limiting factor. While it's going up, there's a slope, light intensity is a limiting factor. If you increase it, it will increase the rate of photosynthesis. So if we move across to the carbon dioxide graph here, you can see it's a very similar shape. And so therefore the pattern's exactly the same. While it's going up, carbon dioxide is a limiting factor, but when it gets to here and it goes straight from that point there, carbon dioxide is no longer a limiting factor. So if we look at these two together, we can see that we have a reason why if we were devising a farm or something, for example, there's no point going beyond that light intensity. There's no point going beyond that carbon dioxide level. So anything more will be a waste of money. Now, the third graph that we need to know is temperature. And this mimics the enzyme profile for temperature. So as we increase the temperature here, we increase the kinetic energy of the particles. And so we remember that an active site fits exactly the substrate. And so because there are hundreds and billions of substrates and enzyme molecules, remember they're all microscopic, they are constantly whizzing around and they will collide, substrate will collide with active site randomly. So if we increase the temperature, we increase the kinetic energy, we increase the speed of the particles, and there's more collisions between active site and substrate. That goes up and up and up and up and up until we hit the optimum temperature. Okay, the optimum temperature is the temperature at which there is the highest activity. After that, we know some key terms. The enzyme denatures because the active site changes shape. So if we had an active site like the one I just showed you, imagine if it turns to that shape, the substrate no longer fits it exactly, and therefore the enzyme rate will go down until a certain temperature here, where the activity is 0 and the enzyme is completely denatured. Remember that pH can also change the active site shape, and that's why digestive enzymes don't work in the various places. So there are 3 graphs. Now we can then look at quite an interesting graph here, which combines a few of these things. So this is a light intensity graph. So we can see as the light intensity increases here, the rate of photosynthesis goes up, as you'd expect, until that point there. Okay, so what we can say is that in this graph, these here are different concentration and temperatures. Okay, so you can see that if you had this light intensity at this carbon dioxide concentration and lower temperature, we get that amount of photosynthesis. If you increase the light intensity and you increase the carbon dioxide concentration, you're going to get a higher one. And if you again go to there with these higher, even higher, you can see we increase the rate of photosynthesis. Now, once we get to this point here, if we did have higher CO2 concentration and temperature, you know, if it was to be up here, you know that because the light intensity can only go to this amount, then this increase must be due to the temperature or carbon dioxide. Because if it's gone up, it must be either carbon dioxide or temperature that's the limiting factor, and not light intensity, because light intensity finishes here. Okay, and so that's a bit about the limiting factors. The last limiting factor, which is minerals, is for chlorophyll, which is the pigment that we have in chloroplasts to do photosynthesis. So if a plant didn't have magnesium ions, for example, which are used in chlorophyll, it wouldn't be able to do photosynthesis. This is why we need to give plants fertilizers. Anyway, I hope that helps.